About a century ago, at the time of Louis Pasteur and Robert Koch, the word virus was generally referred to as a poison. The present notion of virus is entirely different. Now, viruses are recognized as particles of nucleic acid, often with a protein code. They replicate in living cells and cause many diseases, such as influenza, hepatitis, smallpox, and AIDS. The branch which deals with the study of viruses is known as virology. The word virus is derived from Latin word venom, meaning poisonous fluid. It can be defined as non-cellular infectious entities, which contain either RNA or DNA, normally encased in proteinaceous coat, and reproduce only in living cells. Viruses utilize the biosynthetic machinery of the host for its own synthesis and then transfer themselves efficiently to other cells. Some viral diseases have been known for centuries. In fact, the first infectious disease against which effective method of prevention was developed was a viral disease. In 1796, Edward Jenner first vaccinated an 8-year-old boy with a material removed from cowpox lesion on the hand of a milkmaid. After 6 weeks, the boy was inoculated with the pus from a smallpox victim, but he did not develop the disease. Jenner used material for vaccination from cowpox lesions and successfully vaccinated 23 persons. As the material he used was obtained from cow, Latin vacca. Later, the term vaccination was used by Louis Pasteur for inoculation against disease. In 1884, one of the Pasteur's associates, Charles Chamberlain, found that bacteria cannot pass through porcelain filters, while agents responsible for rabies, a disease which is transferred to humans by bites of rabid dogs, foxes, cats, bats, and other animals, can pass through these filters. As in those days, the word virus was loosely used to describe any toxic substance that caused disease. Those unseen, filterable agents of the disease were described as filterable viruses. In 1892, Ivanovsky discovered that the agent which caused tobacco mosaic disease was filterable. He obtained bacteria-free filtrate from ground-up infected plants and placed it on healthy leaves of tobacco. He observed that filtrate produced the disease in healthy plants. Presence of similar filter-passing ultramicroscopic agents was seen in the victims of many diseases, including foot and mouth disease in 1898. The filterable agents were first purified in 1935 when Stanley was successful in crystallizing the tobacco mosaic virus. Chemical analysis of these particles showed that they contained only nucleic acid and proteins. This suggested that unlike other forms, viruses are of simple chemical composition. Characteristics: viruses are extremely small infectious agents which can only be seen under an electron microscope. They range in size from 250 nanometer of pox viruses to 20 nanometer of parvoviruses. They are 10 to 1000 times smaller than most bacteria, so they can pass through the pores of the filter from which the bacteria cannot pass. Viruses cannot be grown on artificial media. They can reproduce only in the animals and plant cells or in the microorganisms, where they reproduce by replication, a process by which many copies or replicas of viruses are formed. Thus, the viruses are obligate intracellular parasites. Viruses lack metabolic machinery for the synthesis of their own nucleic acid and protein. They depend on host cells to carry out these vital functions. During reproduction in the host cells, viruses may cause disease. All viruses are generally resistant to broad range of available antibiotics such as penicillin, streptomycin, and others. The complete, mature, and infectious particle is known as virion. The virions are composed of a central core of nucleic acid, either DNA or RNA, which is also known as genome, and is surrounded by protein coat, the capsid. Capsid gives definite shape to virion. It is made up of protein subunits known as capsomeres. The number of capsomeres is characteristics of a particular virus. For example, 162 capsomeres are present in a capsid of herpes virus and 252 in capsid of adenovirus, which cause some common colds. In some animal viruses, the nucleocapsid, that is, nucleic acid and capsid, is covered by another membrane derived from the host, the envelope. Non-enveloped viruses are known as naked virions. Animal and plant viruses may be polyhedron, having many sides, helical or spiral. Edges occur in two structural forms, having cubical or helical symmetry. In general appearance, cubical phages are regular solid or icosahedral, having 20 faces, and helical phages are rod-shaped. Many phages consist of head and tail. In those cases, heads are polyhedral, but the tails are rod-shaped. Classification of viruses. Virus morphology and nucleic acid properties are most important for classifying plant, animal, and bacterial viruses. The genetic material may be DNA or RNA naked, enveloped, or complex. On the basis of morphology, viruses are classified into rod-shaped, TMV, spherical, polioviruses, and tadpole-like bacteriophages.